I would like to express my appreciation to be invited to this truly exciting meeting. I think I have learned a lot of chemistry in the last few days. And I also like to congratulate all the senior leadership in this country to establish and maintain such high standards of research. The trend is running. I would expect many exciting things to come. And so in the next few minutes, I'd like to share with you the questions. What are the opportunities and challenges for cryo-EM to address the biological question in the context of chemistry? So first of all, what is cryo-electron microscopy? The last speaker laid out very nicely about all the development of high-resolution imaging for material science. So what I want to do is to show you this workflow for cryo-electron microscopy for biological specimens. As the chairman pointed out, it's important to prepare good specimens. In these days, all the molecular biochemistry laboratory can prepare relatively good specimens, but a lot still yet to be done because some of the specimens are very labile. And once the purified material is formed, like in crystallography lab, the first step we do is to freeze the specimen. And these freezing methods were first invented by Bob Glaser, University of California, Berkeley, in the mid-70s, and subsequently refined by Jacques Dubuisset to embed the specimens in amorphous ice, and Jacques was won the Nobel Prize last year for these contributions. And so in the frozen hydrated specimen, then put into this electron microscope, similar to the microscope you just heard a while ago. And since all the biological specimens, unlike the non-organic material, they're extremely beam sensitive. So there are two ways to reduce the damage before we get the pictures. First is we use a very low dosage. In such a way, we zap the picture before the specimens are damaged. What I mean by damage is chemical bonds broken, atoms are moved around and they get sucked into the vacuums. And then the second method in reducing the damage effect is to keep at low temperature. So even if damaged things don't move, you can still see the remnants in the electron beam. So this is a typical images of a low dose images of the frozen hydrated specimen for electron microscope. All these are projection images. I cannot show the images we saw a while ago, where high contrast with every atoms from atomic number 1 to higher numbers. These are actually a biological nanomachines. And so in these projection images, the next step is to do data processing. This data processing was pioneered by Aaron Krug, who laid out all the mathematical principles in the early 70s to go from a two-dimensional projection images to three-dimensional map. So what we're calculating here is a density map. In a chemical term, is actually a Coulomb potential functions. So from this map, if the resolution is good enough, then we can build the model of individual molecules. And once this model is done, and in fact, we now have very rigorous procedure to validate how correct the model is, and subsequently, all the published structures now deposited in the protein data bank and electron microscope database. So now, where is this field right now? So here is the statistic in the last 10 years of all these cryo-EM maps and structure deposited in the public database, and now we reached to about 6,000 structures has been deposited. Now, among these, the resolution of all these cryo-EM structures vary from 100 angstrom to around 2 angstroms. Now, in the last few years, cryo-EM has been picked up as one of the most exciting biochemical and biophysical methods, is because that in the last few years, there are lots of structures are done at the resolution between 2 and 5 angstrom. At these regions, lots of biochemists are excited because many important biochemical questions can be answered. And in fact, such technique was done early in the 2008, and I did this about 10 years ago. And the reason that all the sudden rise is because the development of the detectors. One of the most exciting development that this resolution realization in getting the structures is the detector. I'll make some comment in the next slides. So now, so in order to demonstrate what we can learn about from images to the density map in terms of chemistry, I like to use one particular example to illustrate how this can be done. So this is a well-studied bacteriophage system. Bacteriophage is a virus that infects bacteria. So the genetics and biochemistry is done for decades ago to understand how the gene products are being assembled into, I call, biological buckyball. And then subsequently, in one of the 12 vertices, they are protein apparatus so that the DNA can be encapsulated. Finally, a tail is attached in such a way you can assemble. So the first part of my talk is to show you how we can get a structures of these mature phages, and subsequently how the phages before the DNA come in. Finally, how we can visualize phages are assembled inside a bacterial cell. So here, I'm going to show you an image of these bacteriophages, which is around 700 angstrom in diameter. Now, this is almost an invisible image because this is only the first frame of the image. And now what I'm showing you is this multiple frame, which collected by these direct detectors, which is a very high frame rate. So by adding up all the different subsequent successive frame, we can see this particle much better contrast. More frames are being added together, the contrasts are getting better and better. Now, the value of these cameras are twofold, because the high frame rates, we can align from frames to frame, because when the beam hits the specimen tend to move, and you can see if you want to reconstruct me, when I'm moving, you only get a fuzzy image. But if you take picture fast enough, you can actually align my picture one at a time to enhance the contrast, number one. Number two, Professor Urban showed this modulation transfer of any detector going down here very quickly, and this particular camera actually doesn't go down that quickly, actually go down very slowly, so that all the high frequency information can still be maintained in those images. So as you can see, this number of frames, there are 23 frames added together, and these images are still very noisy. So what we do, these images look different from these. They are basically the same objects, but they look different because they are oriented differently in these matrix amorphous eyes. So the next step of the image processing is actually to recognize what is the orientations of these particles. By combining something like 20,000 particles, we are able to generate this map at 3.3 angstrom resolution. So now, the 3.3 angstrom, what can you see? So as I said, this is a biological buckyball. There are 60 asymmetric units. And what I'm assuming is one asymmetric unit, which is one hexon and then one penton molecules. And these are the density map. I colored it with a color pen. And so each color represents one polypeptide. You can see these polypeptide red on top of the red one, these tucked below the green one. And it turned out there are 7 protein polypeptides make up one asymmetric unit. Each of these 7 polypeptides make up the same chemical contents. Now, since they are situated in a different geometrical environments, as a result of that, even though they are of chemically identical, structurally they don't have to be identical. That's the fundamental quasi-equivalent theory proposed by Alan Cook and Don Casper decades ago. So in fact, what we do is computationally extract each of the polypeptides in one of the color, and then we can build an atomic model from each of the subunits. As I point out, there are 7 protein polypeptides, so we actually build 7 models from the density, and it turned out when we superimpose all these 7 models together, they are not identical as expected, and in fact the deviations in the loop area and also in the N-terminal area. So now, in getting in that point, we see not only how this long spaghetti protein folded in 3 dimensions, we can actually see each individual building block, namely the amino acids that make up these proteins. So here is the model in one of the protein subunit, and from there is a zoomed-in view of all the side chain density. As you can see, that all the side chain density fit the model quite nicely. This long helix, short helix, a loop, another loop, and beta strands are clearly resolved. So you can see, actually, the model we build actually agree with the experimental observation, namely the density of the potential functions of the objects. And so we build this not only for one protein, but 7 proteins in each of these asymmetric units. The rest of the other 59 asymmetrically look just like the one we built in here. So now, once you get this detailed, we call tertiary structure of the proteins that make up this biological buckyball, and what do we learn from here? So what we learn, the first question is, how do these proteins are being held together to form these stable nano particles? And so what we do is now I'm showing you a movie of 3 asymmetric units and see what are the interaction between the subunit from one asymmetric unit to the subunit of the next asymmetric unit. So here in this movie, I depicted one asymmetric in green, another in blue, another in red. I'm looking now, zooming in the interface between the green and the blue. So this model facing outside is inside. So what we discover in our structure is this N-termini actually forming a beta strand. The distances between this N-termini and the other termini is actually at the distance of hydrogen bonding. That means they carry 2 beta strands across from 2 different proteins. So I think that answers the first question, is that how the asymmetric units are held together through this hydrogen bond in the N-termini of the subunit in forming around these 3-fold locations. Now, the next question we ask is, how this protein being held together within each of the hexons? So the next movie I'm going to show you is looking at the structural, the protein-protein interactions between the adjacent subunit within a hexon. So you can see here, I'm showing you from outside this model, and then the one looking from inside. And then I also annotated or displayed the density of the key amino acids we believe are important in the action. It turned out there are about 8 Shaw bridges between any adjacent subunit. So, and in fact, this, in addition to hydrophobic interactions, as we heard from the talk by Dr. Whiteside earlier this week, there are lots of protein-protein reactions, and in here are the Shaw bridges we believe are important in maintaining the subunits within an hexons. So that is the thing we learned immediately from these kinds of analysis with this 3.3 angstrom density map on this mature virus. Now, what I'm showing you only the protein captured cell. And then now I need to move to the next story is about the how it looks before the DNA is encapsulated. So because these types of virus particle is first we form a nanocell, a icosahedral cell, and before the DNA goes in. So we then did the next experiment, which was we call procapsid, and that is a capsid particle which has a slightly smaller diameter. It's 600 angstrom rather than the 700 angstrom I saw in the last few slides. And you can see in these 2 pictures here, in the procapsid without DNA, they're more circular, they are smaller in diameter, but in the virion, they are angular and also about 100 angstrom bigger diameter. The chemistry question is, how does this expansion occur upon the entrance of the DNA? Okay, so because there's no detailed chemistry is done, it's basically a mechanical process. So now, so we then did the same experiment by doing the reconstruction from the density like this. So this is the procapsid. We did it at a 3.8 angstroms, and as expected, it's much smaller than the mature virion. Then the resolution is good enough in such a way we can extract each of the asymmetric unit from the procapsid and build the model. So here is the model shown in the procapsid in cyan, and you can see the topology of the polypeptide is very, very similar, but the detail is different. One obvious one is this N-termini. This one is sticking out this way. This is folding in at the back of this helix, and then also the loops are different. Different loops is different. So from these two models, it's quite obvious that there are substantial tertiary structural change when the capsids are transformed from its procapsid form into its mature form upon the entrance of the genome. So now the next movie, I'm showing you the views about one asymmetric between the two capsids. So here is the one asymmetric unit of the procapsid. You can see here there is an opening in this axomeric subunit arrangement. Now I'm morphing this from this procapsid to mature virion. Now, I want to show this is an interpolation because I have only two structures, procapsid and mature virion. So this morphing is an interpolation, it's not real trajectory. As you can see in the mature virion, there are much intimate interaction between the subunit. Also, the whole being closed up. It's not surprising because after the DNA is come in, something has to be closed up. Otherwise, the enzyme in the cell will digest up all the DNA. So actually it makes sense that actually a large conformational change. Now, so this is a kind of top view, and then I'm showing you in the side view. And in the side view, it's interesting enough that actually when this is the mature virion, the cell actually gets thinner. I said that the capsid diameter get expanded, but the cell protein that form the capsid actually get thinner. So this expansion in bigger diameter and get thinner is actually involves a multiple conformational changes of the polypeptide, not just a hinge motion or a domain motion in many other proteins you experience. Okay, so this is the second story I told you, that from the cryo-EM, we can understand the mechanisms, how the particle can transform its structure from a procapsid stage to a mature stage. Now, in the first cartoon diagram, I didn't get a chance to point out, when you form this procapsid, before the DNA comes in, there's other scaffolding protein to help to build this procapsid. Then the question is, where is this scaffolding protein? So here is the procapsid in the outside view, and here in the, I cut it off from a computer knife, and we can see and highlight some of the density which cannot be accounted for by the capsid protein. And it turned out they are 1-to-1 copy of these red proteins to each of the 420 capsid proteins. So these are the scaffolding protein. It turned out by examining closely the scaffolding protein exactly in contact with the N-termini. As you remember in the movie, the N-termini move. And the reason that move in the maturation, because when the DNA comes in, this scaffolding protein has to go out because there's no space for it. It's only a certain room inside the capsid particle that can either hold the scaffolding protein or the new DNA coming in. So, and in fact, some of these holes you see in the procapsid, the DNA only coming into one of the 12 vertices, but the scaffolding protein actually getting out from all the 60 hexagon subunits. Now, since all the phages you may recall in your high school biology textbook, it actually have a tail. So far, I only show you the capsid, and where is the tail, which is the business end of that. So in this case, we have to relax our image reconstruction protocol, so assume no symmetry in such a way that we see also the tail protein. You can see actually very nice cross-sectional view in these reconstructions. And in here, and in fact, I can color it, and there are 5 more sets of protein in one of these 12 vertices, and each of the protein have different numbers in the parentheses. So I think we can resolve them all at once. So in other words, in this kind of reconstruction, I don't have time to talk about, we can see the molecular interactions, how the 12 proteins at this 5-fold location, the symmetry mismatch, how this symmetry mismatch is accomplished, they all can be resolved in here. So I didn't have the chance to talk about the DNA in here because we have not been able to see the DNA convergence, because it turned out every particle, the DNA package is somewhat differently from other particles. So we cannot do averaging method as we did on the capsid proteins. Now, so this is what I have shown you, how we can use the cryo-EM in highly purified virus particles and look at in the cryo-EM to depict its near atomic resolution around 3 angstrom resolution. Now the question is exciting to the biology. How would this virus particle look like structure-wise inside the cell? So the next few slides, I'm going to show you our approach, how we can look inside the cell to look at the virus assembly process. So here is a microscope that now, if the particular cell I'm talking about is a cyanobacteria, it's a photosynthetic bacteria. And so when you look at the cell, this is around 0.7 angstrom, you can't see anything because they are very thick, you know, about 0.7 micron. Now, so what the specimen area is in here, but then we actually modify the microscope, put a face plate, which was suggested in the optical microscope by Sunney. Is the same idea that we put a face plate, which is actually a thin carbon film with a hole in the middle, in such a way that the scattered wave and the unscattered wave are by a pi over the 2 phase shift, in such a way that after you put the face plate, the contrast substantially improved compared without the face plate. So I think this image contrast enhancement is really substantial. So now, as we know that every cell looks different from any other cell, so we cannot do the averaging like in the virus particle. We average tens of thousands of them. So now in the cell, we need to do the reconstruction 1 cell at a time. So in doing so, and here I'm showing you a reconstruction of one example of one of these cyanophages, which has been bacteria has been infected by phages. So you can see there are a lot of features. Keep in mind, this is no net, no stand, no metal stand, no fixative, completely frozen hydrated. So there's a lot of features in here in this 0.7 micron wide diameter photosynthetic bacteria. And so now, in here is so much information, and if we need to look at it, do it by hand, it would take about more than 10 days to color code which one is which feature in the cell. And so, but recently we actually developed a deep learning procedure. We can do it computationally in a couple hours. So what I do show you is an old annotation by hand. This is the cell envelopes in brown color, the firecon membrane in green color, and then this is the carboxysome that hold up all the Robisco, the fixed carbon dioxide, and then here the vesicles, here as the infectant phage particle, and in here there are new particle form, and the purple color are the newly synthesized, the baby phages that are born or synthesized, assembled in the bacteria. I'm showing you in here this particular area, and there's a plenary ribosome, the plenary ribosome in the cell. Thank you. Yep. And so, and in here, the one interesting aspect of this thing is this particular cell, these particular phages. And it turned out these phages very unusual. It not only have tail, but it also at its opposite vertex, it also has protruding density we call a horn. So one of the questions is, does this horn has anything to do with the infections? But by looking at this picture, we say no, because the tail is still what the tail does, which is penetrate through the cell wall. The horn is sticking out on the other side. So that suggests the horn must involve in a different kind of biological functions. And then let's keep doing the movie. And then now in here, and you can see actually this cell is slightly leaking, so these polyribosomes are actually leaking out the cell wall. Now, so we can do this not only one experiment, we can do multiple experiments as most of the biologists tend to do. So we can have a tomogram of it without any phage infection, the early phage infection. In fact, this bacteria are in the process of dividing, and then this one is in the intermediate stage infection, this is the late stage infection. So in such a way, that's a lot of newly synthesized phagous particles are in there. So then from here, from this kind of tomogram, we are not only looking at it, admiring it, we actually do another step of data analysis. The question is, all these purple color particles, do they really look structurally identical or not? That's the questions. So computationally, we find, no, we were able to find 5 classes of particles. One of them has both the tail and the horn, another has only the tail. The other one class of particle, no tail, no horn. The other one looks angular, but also empty inside without the DNA. The last one is actually a kind of roundish without any dent. So we believe this is the procapsid with the scaffolding protein. So based on this, we actually see one of in here, which we believe is the ATPase in such a way that will help the DNA package into the virus because it needs energy. So I think, in fact, when we do biochemistry, we were able to only purify this biochemically. We were not able to purify the rest. So one important take-home message is, in biochemistry, you can only do so far. You can do what you can possibly do. But a lot of these molecular machines or intermediate in the biological process, they are just so labile or so short-lived that you cannot even biochemically purify it. So I think this is a step using cryo-electron tomography, just looking at the tomogram, doing the data analysis, we can capture different types of intermediates during a biological process. In this case, is the virus assembly inside the cell. So you can imagine that many other macromolecular machines inside the cell, sometimes you can purify to do very high resolution structure, and other time you just can't do it. My suggestion, just looking inside the cell, and then you do the chemistry if you have a high enough resolutions. And so the other thing is, particularly this enzyme, we can never been able to visualize it in purified stuff. Okay, so in here, we can actually map out all the assembly pathway. So in the old day, you need to do genetic, very complicated biochemistry. Right now, you just image it, and then you see them all. So in the next 2 or 3 minutes, 2 minutes, maybe I'll say what the future is. So now, since what I show you in the chemical criterion is really low resolution, only 3 is enzyme resolution. And so the question is, can we go at a high resolution? The answer is yes, because it's very important for drug design, enzyme engineering, nanoparticle, and synthetic chemistry. So how good can we go now? So, so with some of the modern microscope, we can go down in our hands to 2.4 angstrom. This is another nanomachines that we involve in the protein folding. We can begin to see water, the ATP, aluminum fluoride, and magnesium atoms. So I think that is closer to an area that we can look at a drug ligand bind to a molecular machine like a chaperonant. And so this is a soluble proteins. And how about membrane protein, which is a many drug target? So this is one of the V0 from proton channel. It has 15 protein. We can resolve them all at 2.7 angstrom. We can begin to see the lipid moiety in addition to the protein as moiety as well. So that's truly really exciting. We can see all the ligands and other bind to a membrane protein of these sorts. And lastly is what happens if you undergo a biochemical reactions. So here is an example that GrowEL with the cofactor GrowES with the enzyme with the ATP. There are 5 reagents in the reaction product. You don't expect seeing only one product. You will see at least the reagent plus some intermediate. So that's true. We can see the bullet shape. You can see the American football shape. And by data processing, we actually analyze these football-shaped particles in these mixtures. We see some is empty. One has actually density inside. So that means the take-home message is now we can work with molecules that are not highly purified. Unlike crystallography, you need to purify to 100% before you can do the structure analysis. We can now do highly heterogeneous materials and then do image processing. The one important take-home message is there are more sophisticated image processing need to be developed. And final example is now what happens you're looking at a kind of mammalian cell. So this is I think the trend is, can we look at the cell in a normal state and pathogenic state? Where is the human health issues? I want to show you a axon, a neuron from a mouse model of Huntington disease. So this is, forget about the hole in here, but I'm just showing you all the features. And there's a larger feature. This is a holy grid, so this is an artifact in the preparation. But here is the microtubule in red. This is the actin filament in green, and there's a lot of ribosomes in here. And then, in addition, there are actually chaperone, and we can detect as well. So this is, I think, truly exciting because now you can do the deep learning from a tomogram to this color picture in actually an hour. Okay, so then from there we can actually segment different regions to do analysis. And finally, one more example is now, instead of a neuron from a mouse, and we can actually using the stem cell derived from a patient of Huntington disease with this Q repeat of 77. And for years, I want to see how this protein aggregate in the cell looks like. It's similar to the Alzheimer that many of us actually are interested in having that problem solved. So I kind of like to see how this protein aggregate look inside the cell. The last movie I'm going to show you is now we succeed. This was just obtained a couple of weeks ago. It's really exciting. We can now begin to see all these bundles of aggregates. So now the exciting aspect is we want to look at the patients of different Q repeat because that would dictate whether the disease when will be manifested. And also we want to see how these aggregates are being surrounded by what other cellular structures so that in such a way we can develop therapeutic solution to it. And finally, I think I have shown you that the cryo-electron microscopy can span the range from 80 angstrom to 2 angstroms, from a whole molecular cell to organelle to molecular machine as small as a piece of RNA molecule, and we can begin to see water and metals. And finally, I'd like to acknowledge all my people in the lab, the Bacteriophage Cell Project in collaboration with Professor King at MIT, and then the Channel Project with Professor Wilkins and Syracuse, and the Neuron Project with Professor William Mobley in San Diego, and Leslie Thompson in Irvine on this neuronal cell. Thank you very much.
Thank you, Dr. Chiu. Wonderful presentation. After the first wonderful presentation, questions, comments? Everybody's overwhelmed. All right? One here.
Is there some biological significance to the fact that the DNA in the capsid doesn't have an explicit structure, whereas, of course, the capsid does?
Right. Okay, so in these particular bacteriophages, each of the DNA apparently doesn't have a specific interactions with the capsid protein in this particular case. And in fact, there are recent studies that in other phages, particularly the double-strand RNA viruses, we have been able to see the double-strand RNA that attached to it. So if I may, I can show you one quick slide of that. Then we can actually see the RNA molecule that are actually quite ordered in such a way. Every of these RNA virus, the DNA are arranged the same way. And in fact, we can actually see the RNA polymerase at 10 out of the 12 vertices as well. So those are possible.
But is there a reason for that? Biology likes precision. We see precision everywhere. Why, with the DNA, isn't there that precision there?
Okay, that's a good question. Because the assembly process is quite different in the case of this RNA virus, because actually the RNA are packaged together with the capsid protein when it's formed. But in the case of the DNA phages, the DNA is just encapsulated inside afterwards. So the process of assembly is different. And in this case, I think the assembly of the capsid protein also probably influenced by the genome as well. In the case of the DNA, it's just a container, basically. Okay. In this case, there's a polymerase in the vertices. In fact, there's 9 segments of the RNA, but it has 10 pieces of this polymerase. So that probably is not related to the enzymatic activity, but has to do with the stability of the capsid.
All right,
That's
one more?
Question right there.
That will be the last question.
In your GrowEL, GrowES assembly, could you watch the reaction when the GrowEL come on to the GrowES and the protein?
I would love to see that. I would love to see it in the cell. I haven't got that yet. But the next action is looking at the bacteria, and then particularly some systems that overexpress the GrowEL and GrowES to see that process. Because there's still a lot of controversy whether the GrowEL and GrowES for the American football or a bullet shapes of particles. So those are still remain controversy. So the solution is actually cryo-EM tomography.